thank you for letting me give you a glimpse of some of my research that is in support of NOAA's Omics strategy. This strategy was released last week at the White House and was initiated and catalyzed here at AOML. Omics is actually a suite of technologies. For example, we can use DNA or RNA to describe the organisms that are in a community, what they're doing, and how they might adapt to changing conditions. The omics revolution has been driven by sequencing technology that continues to provide us more data for less information. More data for less money. And so one of the things we're trying to do is make sure that we actually can get information out of that data, which is the difficult part and one of the things we're working on here at AOML to ensure that we get actionable information from that data. And it's giving us technologies that are less expensive and are faster than traditional technologies. And AOML is applying these techniques to national priorities, including enhancing fisheries, combating harmful algal blooms, and working to make our sample processing more efficient. All of this is to support the blue economy, which is worth over $300 billion per year to the US. And we're working to sustain and grow the benefits we receive from our oceans and Great Lakes. One area of discovery is the ocean microbiome. That's your viruses and bacteria and phytoplankton that form the base of the food web and thus support food production and do other important tasks like decompose pollutants and recycle nutrients. AOML is working to integrate omics technologies into our observing programs, for example, Cal Coffee, which is one of the oldest programs. This project is in collaboration across NOAA line offices and between academia and industry. And we're finding that omics information is data rich. Compared to a chlorophyll value, which gives you a single number, an omics profile gives you a community structure, and that changes in ways that might not be apparent by chlorophyll alone. We've been doing this work during rapidly changing conditions. For example, the California current has been having a heat anomaly, and this data has been transferred to NOAA modelers, and they're looking at connections between the microbiome and higher trophic levels, particularly for fish, and looking for new ecosystem indicators. In addition to bacteria and phytoplankton, environmental DNA can let you detect higher trophic levels, such as fish or marine mammals. This approach has a number of benefits, and it's the reason it's such an active area of research. One of the areas is detecting fish, and I've been working with our fisheries colleagues for qPCR for fish, and also sequencing or metabarcoding of ichthyoplankton, including historical archives that are in ethanol, so that we don't have to destroy the samples. We've been applying these techniques to look at protected species. Turtle sampling is stressful on the animal and very intensive, labor intensive. And we have some really encouraging results from this preliminary study, and we weren't sure if it would work because of the hard shell. We're working internationally. The omics strategy calls for expanding our partnerships. A new area is the marine microbiome roadmap for the Atlantic between Canada, the US, and EU that will be rolled out in 2020, February. And we've been working with Norway in a bilateral, also working with National Marine Fisheries to explore a potential fishery in the mesopelagic using eDNA technologies. One of the great things about eDNA is it is amenable to in situ and automated platforms, and we've been working with Mimbari to push these technologies that have omics capabilities and to see if that can enhance our ability to observe the ocean in a more cost-effective manner. Our engineering progress in the last few years, we've moved from a prototype instrument that can sample only 5 samples to one that can sample 60 samples. And in addition to having this prototype that can sample for omics analysis in the Great Lakes, that prototype we're using actually has real-time toxin detection for Hab toxin. And that ability exists within the payload of the autonomous instrument. Now, all of this success is predicated on having the expertise and tools needed to do this work. And AOML is dedicated into enhancing our workforce and our infrastructure. And I'm going to refer you to a poster by Luke Thompson so you can get some examples of our bioinformatics pipelines, and those are available on GitHub.